Hello, Biology 107 fans. We left off in class talking about the two theories that really unify all of biology, the cell theory and the theory of evolution. And we were going to talk about how artificial selection instead of natural selection can help us understand the process of natural selection. So what is artificial selection? Artificial selection is the process by which we actually take two organisms and mate them together, and we're manipulating the gene pool in a particular way that allows us to pick and choose genes that we want to enhance in the following generations. And we began to talk about this in class. We were talking about dogs, purebred dogs, especially German Shepherds, have now been bred together. So we breed together siblings from litters that are particularly nice, meaning that they have the genes, they're phenotypically looking like the dogs we want, and we breed them together, and then we breed their children together, etc., until we actually have problems in the gene pool because you end up with things that are not necessarily good for the animals, such as the hip dysplasia. Some dogs are prone to cancer. Some dogs have knee problems, and so it can be a little bit of a double-edged sword. Some things are good, some things are bad. Let's look at this example. This is looking at taking the organism corn, very simple organism, right? So you take some corn and you say, I want to breed together the corn that's best for me. So you breed together corn that has higher levels of protein per kernel than lower levels. And so over the course of many generations, what you see here is, so as you start breeding these corn together, higher and higher levels, right, you end up at a level of protein. So the average percent protein per kernel of corn increases. And is that a good thing or is that a bad thing? Well, that's really good because we need high protein content for populations that actually don't have very much food. So this is actually a really great thing to do. So this is really a genetically modified organism. A GMO food. And it's genetically modified by us actually breeding together corn that has high levels of protein content per kernel. So artificial selection, right, is done. We do this all the time. So humans can do this by selecting what we consider desirable traits within what we call a domestic population, something that we are breeding. Evolution is going to occur in any species. And evolution is really talking about, right, the heritable changes in genes. So heritable meaning it's passed down, and the variation occurs in genes. And from one generation to the next, right, successful reproduction leads to evolutionary changes. And those variations can occur either by natural selection, which means that's just two animals getting together, or two corns getting together, or two whatever's getting together, right? Or through human manipulation using artificial selection. We began talking a little bit about fitness, and fitness isn't, oh, you're really fit, you look great, you have a lot of muscles. Fitness is all about the ability to produce offspring. And it's not just the number, right? It's not just the number of offspring, but it's the number and fitness of those offspring. Hey, remember, if you have 500 babies, but all the babies die immediately, that's not very fit. So we're talking about fitness And how many, right, with surviving offspring do you make? What's the process of adaptation? Right, adaptation is when a trait increases the fitness of an individual, which allows it to increase the number of offspring it produces. So what would be an adaptation in an organism? Let's say you have an organism that has sperm that have a little short flagella, right? And that little flagella, it's okay. It can allow this guy to move okay, right? But if you had an adaptation that changed that sperm into a sperm that had a super long flagella, and now those sperm could swim much better, that might actually increase the fitness of an individual. And if that particular person was able to pass down that adaptation in that gene, right, that is going to drive natural selection. Natural selection to the organism that had more fitness, the longer sperm, the longer flagella on the sperm. So both fitness and adaptation drive natural selection. So as I said, these two theories, the cell theory, right, saying that all living organisms are made of cells and that all cells come from pre-existing cells, really ties together with evolution, because if all cells come from pre-existing cells, that suggests this hereditary mechanism. And the theory of evolution is talking about how species change over time due to natural selection of genes that increase fitness. And this is what has allowed us to generate the tree of life. The tree of life takes together these two concepts, and originally the tree of life was based pretty much on visual observations. What organisms look most similar? Which organisms are furthest apart? Which ones could you suspect came from one another? And since all species come from some pre-existing species, you could then start to put them in an order. What this really tells us is that eventually you have to go back to a single common ancestor. And what the heck was that single common ancestor? It was the original cell that was made back when the Earth was being formed, approximately 3.5 billion years ago. Remember, Earth came into being 4.5 billion years ago, so it took 1 billion years for that first common ancestor to form. And since then, what's been happening, so over the last 3.5 billion years, is you have the process of speciation through divergence. Natural selection has allowed for all the different species to emerge over the last 3.5 billion years. Pretty cool stuff, if you ask me. Looking at the phylogenetic tree, right, you can then begin to understand what the lines mean. The lines have meaning to them, right? So down here is where we have this common ancestor. And that common ancestor had heritable material, right? It was a living being. And so it has all 5 of the traits of living organisms. And right here at this node is where the first speciation event occurred. And this was where the bacteria split off from Archea and Eukaryotes. So that was the first speciation event. And if you continue to the right, at this node here, at the node that separates Archea from Eukaryota, that really gives us the separation between, what did we say these guys on the left-hand side are called? These guys are called prokaryotes. And pro meaning before karyotes. And what did we see the karyotes were? A kernel of corn. And that kernel of corn really is talking about the nucleus. So this helps us understand this tree of life. The lines on the tree of life represent the number of changes in genetic material from one speciation event to the next. So if you look, right, if you compare fungi on the top right here to animals, you would actually track down this branch and up this branch, and usually they give you on the tree, they'll say 10 nucleotides changes per 1,000 nucleotides or something. They give you a key, and so you can measure that line and then have a estimate of the number of mutations or differences in their genomes between two different organisms. So the tree of life actually helps us to begin to understand our heredity. You can make your own tree of life with your family, right? You can do Ancestry.com and look to see where your family comes from, and that's looking at these genealogical relationships. But not just within a species can you do that, right? Humans can do that in our species, but we can do that from one species to another. And what that process is called is phylogeny. So we're looking at phylogeny when we're looking at the tree of life. Remember, I told you that originally the way the tree of life was made was based on observations. What did Darwin look at? Darwin looked at finches. He looked at a set of birds that have different beaks. They were all finches, they were all called finches, but each finch was slightly different than another. And so those differences were visualized, and he could make some comparison and say maybe this particular beak evolved from that beak, and this beak evolved from that beak, and so on and so on. But that isn't very specific, and as scientists we want to have a really firm grasp on what those differences mean. And it wasn't until this guy, Carl Woese, began to look at a molecule called RRNA. What does RRNA stand for? Okay, I wrote it down here for you. It's ribosomal ribonucleic acid. And what this is part of the genetic material that you have, right? We know that we all have DNA. And from DNA, you can have the formation of RNA. And there are different forms of RNA, and one of the forms of RNA is rRNA, ribosomal RNA. And this RNA actually is used inside of cells to help make ribosomes, and ribosomes are used to help translate RNA into proteins. Not rRNA, but mRNA. Okay, that's getting too complicated, but let's go back to the rRNA. So the rRNA is made up of nucleotides. And this is where you need to start understanding some of the structural relationships that drive biology. So I'm going to draw for you what an RNA molecule, an RNA nucleotide, looks like. So RNA molecules are made up of a phosphate, a sugar, and a base. And this base is a nitrogenous base. Okay, let's start with the sugar. The sugar is a pentose. What's a pentose? Well, a pentose is a 5-carbon sugar. And the way you draw it, and you do need to be able to draw this, okay, is it starts with an oxygen molecule and goes to a carbon, and that's called the 1 carbon, to a 2nd carbon, the 2 carbon, to a 3rd carbon, 3, to a 4th carbon, here, 4, And this actually links back to the original oxygen. Well, I said it's a pentose, which actually means it's a 5-carbon sugar, and the 5th carbon comes off of the 4th carbon directly up from the ring structure, and this is the 5th carbon. And there are other molecules associated with this structure. Now, I've already done some chemistry right here. Every time I drew a line, I was drawing a covalent bond. And we haven't talked about that yet, but we will. And covalent bonds are very strong. Okay, so we have a 5-carbon sugar, and I told you that there's a phosphate group, and that phosphate group is linked to the 5th carbon. And the way that it works is that phosphate groups are O, P for phosphate, O, the O's are oxygen. And if I just draw one phosphate group here, this phosphate actually needs an oxygen there and an oxygen here. And these oxygens actually have negative charge. So this is the phosphate group, all right here. I've drawn for you this 5-carbon sugar. And then the nitrogenous base actually comes off of this 1st carbon. And I'm not actually good at drawing these nitrogenous bases, and I won't ask you to draw them, but let me tell you what the nitrogenous bases are. They are molecules that are called adenosine, guanine, cytosine, and uracil. A, G, C, and U. And this is specific for RNA. So we're just going to draw a box up here for now, and you can just put in any one of the nitrogenous bases. Let's put an A in there. Okay, well, I've left out some really important features of this nucleotide. So this is a nucleotide. And probably one of the most important features is what is linked to the 2, so we call this the 1' position, the 2' position, the 3' position, the 4' position in the ring structure. On the 2' position on an RNA molecule, RNA stands for ribonucleic acid. And so what it means is that on this carbon there is a hydroxyl group. So on the 2' carbon there's a hydroxyl group. And interestingly, on the 3' carbon there is also a hydroxyl group. Notice you can draw the hydroxyl group OH or HO, it doesn't matter. And this would be the beginnings of the drawing of an RNA nucleotide. It's still missing some atoms, and we're going to talk about how that works in class, but this would get you really started with this. So if you can draw this, hint, 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 that would be really helpful. This might show up in class the next time we meet. So this is an RNA molecule. And what Carl Woese did was he compared strings of RNAs, so RNA molecules. Here in these images, what is shown are the linkage of nucleotides together. And this here, what I'm what I'm pointing to is a string of RNA, and it actually has ends which they haven't drawn on here. And this will become clear to you later. It has a 5' and a 3' end. And so this is how these nucleotides get put together into a string of RNA. And as I said to you, they're what I call 4 flavors, A, C, G, and U, which stands for adenosine, cytosine, guanine, and uracil. And there are distinct sequences of RNA molecules in every organism. So you can look at the sequence here that I've drawn the red arrow to, and you can compare that sequence to the one that I've drawn the blue arrow with, and you can say, all right, so as long as these are exactly going in the same direction, we can say, look, how similar are these two organisms? Well, the first nucleotide's the same, the second's the same. You can just go down the list, you just compare. Oh, look, oh, look, this one is different. So this isn't exactly the same organism, and you can keep going, right? And so how many differences between these two organisms tells you how far apart those two organisms are on the evolutionary tree. Now, here you're only looking at 2, 4, 6, 8 nucleotides, and that's not the number of nucleotides that are in a particular given rRNA. So some of the rRNAs can be up to 2,000 nucleotides long. So what Woese did was he compared the sequences in these rRNAs across all 2,000 nucleotides, and he was able to come up with a really accurate phylogenetic tree that we now have today. So this allows us to see what species are closely related and which species are really far apart. And all he did was compare how many changes from one species to the next in these rRNAs. So if we look at this little table down here, what this is different species. So we have species number 1, 2, 3, 4, 5, 6, 7. And this is just comparing a very short sequence again, right? I can't give you 2,000 nucleotides to look at because that would take you all day, right? But if I gave you a sequence like this, you should very easily be able to look at it and say how many nucleotides are different from species number 1 to species number 2, right? And what would you do? You would look at the first nucleotide and you would say, oh, G in number 1 and G in number 2, that's the same. U in number 1, U in number 2 for the second one, all right? U and U, G and G. Oh, look, we have something different here. So in this one, where I have the red arrow, you have a U and a C, so there's a difference. All right, you go to the next one, it's the same, different. And I'm just looking at comparing organism 1 to organism 2. Okay. Same. So if you're comparing organism 1 with organism number 2, how many nucleotides are different? Okay, 2. So there's 2 nucleotides different between organism 1 and organism 2. What if you compare organism 1 with organism 3? Well, so GG for the first one is the same, UU for the second one, UU, GG. UU is the same, so that one's not different. AA, UU, GU. So there you have one different, right? So this one's different. GG, UU, UU. So that one only has one difference between organism 1 and organism 3. So what could you say about organisms 1, 2, and 3? Well, because there's one difference between 1 and 3, it's more closely related than organisms 1 and 2. And this is what was done to generate the phylogenetic tree. So imagine how this might look on an exam. You might be given a sequence, and I could say which of these organisms are most closely related and which are the furthest apart from one another. And all you have to do is compare the sequence. So once again, I hope what that does is gives you a better understanding of what you're looking at when you look at this phylogenetic tree. This is the phylogenetic tree that Carl Woese was able to generate. And the big difference was that Carl Woese was able to determine that we have 3 domains that are separate from one another. And this was all based on looking at RRNA sequences. Very, very important discovery in biology. So this is allowing us to understand the relationships between one species to the next. The branches on the tree, right, are most closely related when they're all together, and branches that are not right, closely related, suggests that these are distantly related. We're all related. That's the bottom line. You and me, and the dogs and the bacteria, everything that is alive on the planet Earth is related by a single common ancestor. And that single common ancestor was the first cell that had genetic material that was able to reproduce, utilize energy, and able to evolve. So just keep in mind the tree of life, right, has domains, so that's really important, and it was determined based on rRNA, and it was Carl Woese who just did this. He just died a year or two ago. Really important guy. Okay, prokaryotes are before kernels, before nucleus. So bacteria and archaea have no nuclei, whereas the eukaryotes have a nucleus and membrane-bound organelles. So just leave you with this. The nature of science is all about asking questions, and by figuring out proper ways of analyzing those questions. If you can design an experiment that will give you information that will answer these questions, you collect data, you analyze the data, and that allows you to formulate hypotheses. And eventually, if you can have supporting data for hypotheses time after time after time after time after time, and I'm talking about over maybe tens and 20 and 30 and 50 and 100 years, those hypotheses can then become theories. And that's what's happened with the theory of evolution and the theory of cells. All right? So all of science is based on designing experiments, analyzing data, and then modifying hypotheses based on the findings that you get from the data. And that's going to be it for right now. Have a good day, and see you next week.